Let us try and understand glycolysis. Now, glycolysis is a universal pathway. It is also known as the Embden-Meyerhof pathway. So basically what happens is in glycolysis we have a 6 carbon glucose which is split into two 3 carbon products. One is 3 carbon pyruvate and the other is 3 carbon lactate. Now pyruvate is formed when there is oxygen present. So under aerobic conditions you will get pyruvate. Under anaerobic conditions, when we do not have oxygen, then you get formation of lactate. Okay. Now glycolysis occurs in all the cells of the body and it is the only source of energy for the RBCs. Why? Because RBC does not contain mitochondria. The anaerobic glycolysis, which leads to production of lactate, is the major source of energy in the active muscles. Okay. And pyruvate, which is formed from this pyruvate, we also get synthesis of non-essential amino acids. And something else is also formed in glycolysis, which is glycerol, from which we get is triglycerides. So let us look at glycolysis. So this is the pathway that we will try and understand today. Okay. So if you look at the major outline of the pathway, we have is glucose here, which is metabolized and ultimately converted to pyruvate or lactate. So in this process, 2 ATPs are used. Okay, one ATP is used here and the other ATP is used here. So glucose uses 2 ATPs, undergoes phosphorylation, and ultimately we get formation of 3 carbon compounds. Okay, so the 6 carbon compound, which is fructose 1,6 bisphosphate, is broken down into 2 3 carbon compounds. Okay, which is glyceraldehyde 3 phosphate and an isomer of it, which is dihydroxyacetone phosphate. So in other words, we get 2 molecules of glyceraldehyde 3 phosphate. Then each molecule of glyceraldehyde 3 phosphate will lead to formation of 2 ATPs and 1 NADH. Okay, so in this pathway, if you see, glucose is broken down. Initially 2 ATPs are used. Glucose is a 6 carbon compound. It is broken down into 2 3 carbon compounds, and each 3 carbon compound then leads to formation of 2 ATPs and 1 NADH. Okay, so let us look at the pathway. So glucose is first phosphorylated to glucose 6 phosphate. So this phosphate group is donated by the ATP, which is adenosine triphosphate. So it has 3 phosphate groups. So it will donate 1 phosphate group and it itself will get converted to ADP or adenosine diphosphate. Okay, and the glucose gets a phosphate group at position 6. Now the enzymes which bring about this conversion of glucose to glucose 6 phosphate can be either glucokinase or a hexokinase. Okay, both enzymes. But there is a difference depending upon the location. Okay, now if you see hexokinase, it is located in all tissues, whereas glucokinase is located in liver or in the beta cells of the pancreas. Now why is that? The answer lies in the Km value, okay, or the Michaelis-Menten constant. If you look at the Km value of hexokinase, which is very low, and glucokinase is relatively higher. Because of this, the hexokinase has high affinity for glucose. That means even when the blood sugar level is low, okay, even when the blood glucose level is low, then glucose is utilized by the body cells. So that is why hexokinase is present in all tissues of the body. Okay, so that even when the blood glucose levels goes down or is on the lower side, still each cell of the body can undergo glycolysis and can process glucose and produce energy. Okay. On the other hand, glucokinase is present in liver and beta cells of the pancreas. The affinity of glucokinase for glucose is relatively lower. Okay. So this results in action of glucokinase on the glucose when the blood glucose level crosses 100. Okay, so in the liver, glucokinase will be activated when the blood glucose levels cross 100, and as a result the liver converts these glucose into glycogen, which is the storage form. Okay, now this storage form of glucose, this process will occur when there is sufficient amount of glucose in the blood. You do not want glucose getting stored and not being utilized. Okay, so that is why only when the glucose levels in the blood is sufficient, that is, it crosses 100 milligram per deciliter, then the liver starts to convert these glucose into glycogen and store it for emergency purposes. Okay, similarly, the beta cells of the pancreas, they also take up glucose when the blood glucose levels cross 100. So when they take up glucose, in response the beta cells start producing insulin, and insulin has a hypoglycemic effect. That means it lowers down the blood glucose. Okay, so when do we want the blood glucose to be lowered down? When the blood glucose concentration increases. Okay, we do not want, if the blood glucose levels are low itself, we do not want insulin to be released and further lower down the blood glucose. So this is the reason why we have glucokinase present in the beta cells of the pancreas, which have low affinity for glucose. That means they will act on glucose when the blood concentration of glucose is sufficiently higher, more than 100 milligrams per deciliter. And after that, it will start producing insulin. Okay. So glucose gets phosphorylated to glucose 6-phosphate. Okay. Then it is isomerized to fructose 6-phosphate. Then this first fructose 6-phosphate again gets another molecule of phosphate from ATP, which in turn gets converted to ADP and forms is fructose 1,6 bisphosphate. Okay, so here the phosphate group is, one phosphate group is attached at position 1, the other phosphate group is attached at position 6. So two different positions, that is why it is called bisphosphate. Okay, if the phosphate groups would have been attached at the same position, then you would get diphosphate. Okay, so here we have phosphate group which is attached at position 1 and position 6, hence it is fructose 1,6 bisphosphate, and the enzyme is phosphofructokinase. Okay, so this is the rate limiting enzyme of this pathway of glycolysis. Then this fructose 1,6 bisphosphate, it is 6 carbon, it is broken down by aldolase, which is a hydrolytic enzyme. Okay, so here water is not required. In the absence of water, aldolase breaks fructose 1,6 bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which is, these two are isomers of each other. So we can say that we are getting two molecules of glyceraldehyde 3-phosphate which is getting formed. Okay. So the other thing I want to mention is this dihydroxyacetone phosphate. This can further lead to formation of glycerol, which is utilized for triglyceride synthesis. Okay, so from glycolysis we get also glycerol, which is used in triglyceride synthesis. So this is also a link between carbohydrates and fats. So coming back to glyceraldehyde 3-phosphate, so we have two molecules, so we can consider that there are two molecules of glyceraldehyde 3-phosphate which is getting formed. Now each molecule of glyceraldehyde 3-phosphate will undergo dehydrogenase reaction, okay, and addition of a phosphate, inorganic phosphate group. So this dehydrogenase, that is removal of hydrogen. So this hydrogen is accepted by NAD and it forms as NADH plus H positive. Okay. And we have addition of a phosphate group, so this leads to formation of 1,3 bisphosphoglycerate. Then this 1,3 bisphosphoglycerate undergoes loss of this phosphate group. Okay, so ADP is converted to ATP. So the phosphate group which is lost, this phosphate which it had gathered first, so this phosphate here it is lost again, and this phosphate is accepted by ADP and leads to formation of ATP. Okay, so energy is getting formed over here, and the enzyme is 1,3-bisphosphoglycerate kinase. So here the phosphate group which were attached to position 1 is lost. As a result, what is remaining is 3-phosphoglycerate. This 3-phosphoglycerate undergoes isomerization. It forms as 2-phosphoglycerate. Okay. And this 2-phosphoglycerate then is converted to phosphoenol pyruvate with the help of enzyme enolase. Okay. Now this enolase enzyme is the enzyme which is inhibited by fluoride. Okay. So this fluoride is present in our fluoride vacutainers, which we use for blood sugar estimation. Okay. So for blood sugar estimation, we use special type of vacutainers or vials which are called fluoride vacutainers. Why do we use that? Because the fluoride here inhibits the enzyme enolase. Okay, so when this inhibits the enzyme enolase, the glycolysis pathway is inhibited. So when glycolysis pathway is inhibited, consumption of glucose is reduced by the cells. So as a result, whatever is the blood sugar, whatever the blood glucose levels which is present in the blood, the same blood glucose levels we are going to get. Okay, if we do not inhibit this enzyme, in other words, if we do not inhibit glycolysis, then glycolysis will continue in the blood cells, and the blood which is collected will show lower levels of blood glucose. Why? Because the glucose has been utilized by glycolysis. So we want to inhibit glycolysis. So how do we do that? By adding fluoride. So this fluoride, it inhibits enolase enzyme. Okay, now the question arises, how does fluoride inhibit enolase enzyme? So basically fluoride inhibits the enolase magnesium complex. Okay, so that is how fluoride inhibits enolase enzyme. So once a phosphoenol pyruvate is formed, after phosphoenol pyruvate formation, we have pyruvate kinase which acts on it and results in the formation of pyruvate. Here again the phosphate group which is released is accepted by ADP and you get formation of ATP. So under aerobic conditions we get pyruvate formation. However, if it is anaerobic condition, there is absence of oxygen, then pyruvate is converted to lactate. Okay, and in the process NADH is utilized. So NADH is converted to NAD positive. Okay. So this is, let us concentrate over here in anaerobic mechanism. Okay, so I told you under anaerobic conditions pyruvate is converted to lactate, and for this it requires this NADH. Okay, so from where does this NADH comes? This NADH comes from the step where NADH is produced in the glycolysis itself. Okay, so glyceraldehyde 3-phosphate undergoes dehydrogenation to form is 1,3-bisphosphoglycerate. So here the hydrogen which is lost, which was accepted by NAD to form is NADH. Now the same NADH is utilized by lactate dehydrogenase enzyme which converts pyruvate into lactate, and in turn this NADH is converted to NAD. Okay, so this NAD can again be regenerated into NADH. So this pathway becomes self-sufficient. So NADH is being utilized, converted to NAD, NAD then again gets converted to NADH. So under conditions of anaerobic glycolysis, when oxygen is not there, okay, so NADH is utilized and pyruvate is converted to lactate. Okay. So lactate is most commonly formed in the muscles. Okay, so here we have formation of lactate in the muscles. So this lactate again goes to the liver, which is converted to pyruvate by the same enzyme lactate dehydrogenase, and then pyruvate is converted to glucose by the process gluconeogenesis. Okay, that we will study later. Okay. So this was about the glucose metabolism by glycolysis. Now let us look at the energetics of glycolysis. Okay, so I told you before that 2 ATPs were utilized first. Okay, so under anaerobic conditions, when there is lack of oxygen, hexokinase utilizes 1 ATP over here, phosphofructokinase utilizes 1 ATP. So 2 ATPs are used up. Okay. Then we get formation of, from here is glyceraldehyde 3-phosphate. Here you get formation of 1 NADH, and here you get formation of 1 ATP, and here you get formation of 1 ATP. Okay. So at the phosphoglycerate kinase, here you get formation of 1 ATP here from this glyceraldehyde 3-phosphate, which is 3 carbon. So I told you similarly another molecule of glyceraldehyde 3-phosphate is also occurring. So you get is 2 molecules of glyceraldehyde 3-phosphate. So the same pathway is occurring twice. So here instead of getting 1 ATP formation, you get 2 ATP formations. So at the level of 1,3-bisphosphoglycerate kinase, you get 1 ATP plus 1 ATP, so you get is 2 ATPs. Similarly, pyruvate kinase will also give you is 2 ATPs. Okay. Here NADH, you have formation over here. This NADH is getting utilized under conditions of anaerobic glycolysis. Okay, so this NADH is not providing you any energy. So the only energy which you are getting in case of anaerobic glycolysis is from ATPs. So total you get 4 ATPs which are formed. 2 ATPs are utilized at the beginning. So total ATP formation under conditions of anaerobic glycolysis is only 2 ATPs. Now let us look at the aerobic glycolysis. Aerobic glycolysis also, the first part is the same. 2 ATPs are utilized at hexokinase and phosphofructokinase. Here you get formation of NADH. Okay. So because the end product of aerobic glycolysis is pyruvate and not lactate, the NADH which is formed over here does not get utilized by lactate dehydrogenase enzyme. So this NADH goes to the electron transport chain and gives us 2.5 ATPs. Okay, now since there are two NADH formed, so one NADH gives you 2.5, so 2 NADH will give you 5 ATPs. Okay. And the rest part remains the same. Again you get the ATP formation from 1,3-bisphosphoglycerate kinase, 2 ATPs and 2 ATPs from pyruvate kinase. So total you will get is 9 ATPs which are formed. Okay, 5 plus 4 is 9 ATPs. 2 ATPs were utilized at the beginning, so you get is 9 minus 2 is 7 ATPs under conditions of aerobic glycolysis. Okay. Coming finally to the regulation of glycolysis, the main enzyme for glycolysis which regulates glycolysis are hexokinase, glucokinase, phosphofructokinase, which is the rate limiting enzyme, and pyruvate kinase. So hexokinase is inhibited by glucose 6-phosphate, the product of this enzymatic reaction. Glucokinase is activated by insulin, okay, and inhibited by glucagon. This occurs at the genetic levels. Okay, so insulin will stimulate the genes for glucokinase, which leads to more transcription of the enzyme glucokinase. Similarly, glucagon will inhibit the transcription of glucokinase. So this causes the repression of glucokinase genes. Okay. Phosphofructokinase, which is the rate limiting enzyme, phosphofructokinase 1. Okay. So here again insulin activates the phosphofructokinase 1. Okay. How does it activate? It activates by basically by dephosphorylation of phosphofructokinase 2. Okay, so there is, just like this is phosphofructokinase 1, there is also an enzyme which is called phosphofructokinase 2, which converts fructose 6-phosphate to fructose 2,6-bisphosphate. Okay, so insulin stimulates this enzyme, which leads to production of fructose 2,6-bisphosphate. Now this fructose 2,6-bisphosphate allosterically activates phosphofructokinase 1. Okay, so this is how the activation of phosphofructokinase 1 takes place with the help of insulin, phosphofructokinase 2 and fructose 2,6-bisphosphate. Okay, now fructose 6-phosphate is the substrate of phosphofructokinase 1. So if there is increase in the substrate, phosphofructokinase will be activated. Also, AMP is adenosine monophosphate. This is the allosteric activator of phosphofructokinase 1. Okay. Now let us look at glucagon. Glucagon causes inhibition of phosphofructokinase 1 by mechanism which is just the opposite of what is sort of insulin. Okay, so this causes phosphorylation and inhibition of phosphofructokinase 1. Okay. ATP also inhibits phosphofructokinase 1. ATP inhibits by acting at a allosteric site. Okay, high content of ATP inhibits phosphofructokinase 1 allosterically. Okay, low ATP content will activate phosphofructokinase 1. So if you look at the mechanism, phosphofructokinase 1 converts fructose 6 phosphate to fructose 1,6 bisphosphate. Okay, so the phosphate group which is coming, it is coming from the ATP. So here ATP is acting as a substrate, but this occurs at low concentration of ATP. At high concentration of ATP, the ATP is going to inhibit phosphofructokinase 1. And how does it do that? It does by allosterically inhibiting the enzyme. Similarly, citrate, low pH, this also will allosterically inhibit phosphofructokinase 1. Okay. Cyclic AMP is activated upon by a glucagon. So cyclic AMP, when it is formed, it activates protein kinases which phosphorylates the enzyme and inactivates it. Okay, so this is how phosphofructokinase 1 is activated and inhibited, and it is called the rate limiting enzyme of glycolysis. Coming to insulin, coming to pyruvate kinase, again it is activated by insulin and fructose 1,6 bisphosphate. So this insulin, it again acts at two levels. At the genetic level, it increases the transcription of pyruvate kinase, okay, or in other words, causes induction of the gene of pyruvate kinase, so it activates pyruvate kinase. The other mechanism by which insulin activates pyruvate kinase is by dephosphorylation. Okay. Glucagon inhibits pyruvate kinase by phosphorylation with the help of cyclic AMP. Okay. And ATP also inhibits pyruvate kinase by allosteric mechanism. So this is the regulation of glycolysis which takes place. Okay. So hope you have understood. See you next time. Okay. Thank you.